Okay, we are starting off part B, chapter 20, with some figures. And these figures say sometimes things work and sometimes they don't. So let's look at this first figure, annual precipitation as a function of net primary productivity, milligrams of carbon per hectare per year. What we see is what I frequently refer to as the Goldilocks zone of annual precipitation. Not enough, too much, and just right. So if we were to invoke the Goldilocks hypothesis as it relates to net primary productivity in annual precipitation, we'd say that not having enough precipitation limits biological function. In this case, things like for photosynthesis to operate, there's some amount of transpiration that happens and water loss, and so having not enough water is an issue. Having too much water when the porridge is too hot, we have to think about what comes with precipitation. Well, cloud cover, right? So if you're someplace that has 6,000 millimeters of rain, or 8,000 millimeters of rain, it's probably cloudy all of the time. And so that's going to lead to issues, as well as nutrients and being leached from the soil. And then you have to think about when soil gets so wet, it can become hypoxic. You can get sulfate-reducing bacteria, it gets soggy, you get fungal growth, things like that can generally harm productivity. And so there's a happy medium here. Now, temperature, more is better. And so when we look at warmer temperatures, we tend to have, again, a couple covariates with that as well. Typically more sunlight, and sunlight's important for primary productivity. As well as metabolic rates just generally are faster when the temperature is warmer. But you can also have increased respiration rates as well with temperature. And while this can be true, there's a couple instances where that's being altered a bit. There's that example from the tundra of an area that used to be a carbon sink that's now a carbon source because of changing climate. But in general, when it comes to precipitation, we can think about this in a Goldilocks hypothesis. And then with temperature, generally, more is better. So climate influence on net primary productivity can also be indirect, mediated by factors such as nutrient availability. Several approaches are used to determine whether an influence of climate is direct or indirect. So here is an example of above ground net primary productivity, grams per meter squared, in two different ecosystems looking at net primary productivity as a function of precipitation. Sorry, I was just trying to match the colors here and make sure that I know which ones should be which, but trying to match colors here. So let's look at grasslands first, of site-to-site variation. Generally a positive relationship between the two. For the short grass steep year-to-year variation, the net primary productivity in short grass steep systems change in response to annual variation in precipitation. So here it's primarily a year over year. In grassland sites across the central U.S., net primary productivity variation with precipitation was greater across sites than year to year within one site, which they attribute to variation in species composition. So depending on who's there, you'll have an impact in rain this year, an increase in primary productivity this year, or If it's a different species composition, it might be rain this year is more related to growth the next year, that the response might not be immediate depending on the type of vegetation. And we can also look at the role of nitrogen, nutrients, particularly nitrogen is important. Phosphorus is also really important. And so we'll look at a fertilization experiment in wet and dry alpine meadow communities where they added nitrogen N, phosphorus P, and both N and P were added to different plots to see what happened. And this is a pretty interesting one, and so move me here. So we have our dry alpine meadow and our wet alpine meadow. We have our three different, either genera or groups of plants, sedges and grasses, forbs, and this specific sedge here. If we control in the dry meadow, we're adding phosphorus, and there's not a big change. So between the control and the phosphorus, not a huge change. But then when we add just nitrogen, we do see an increase in our above ground biomass, and this is in grams per meter squared. So we could say that phosphorus here is not limiting in natural conditions. So we add phosphorus, not a big increase. We add nitrogen, we get a pretty big increase. That's from like 125 to 210. So that's a good increase in biomass. Now we add nitrogen and phosphorus, we get an even greater increase to almost 400 grams per meter squared. So that's a huge jump. So looking at that, what I would say is phosphorus is not the limiting nutrient in natural conditions compared to the control. Then we add nitrogen, and what likely happens is this increase in productivity, so the difference between this 125 and 210 is the amount of nitrogen limitation. And then this system with nitrogen added is probably phosphorus limited. So then when we add nitrogen and phosphorus, we get a big jump here. So an indication that in its natural state, nitrogen's limiting, and then when we add them both, then we get a big increase in productivity. And in general, we can look at, hopefully, yeah, they put this in order. No, they didn't. Oh man, I don't like that when they do it not in order because our forbs are on top. And Crobetia, the sedge, is on the bottom. You can look at the what's making up this change here. And here Crobetia is increasing a little bit, with nitrogen, it bumps up. Your graminoids increasing a fair amount here. So between control, nitrogen addition, some, but a big jump here. But the biggest change between the control and the nitrogen phosphorus addition is the forbs. Big change there. If I'm reading those colors right, man, I hate colors. But generally there's some increase in all of them. We are favoring forbs are doing much better in the nitrogen phosphorus control. But in general, again, if you see this kind of ecosystem, we're adding phosphorus, it's not phosphorus limited, right? Nitrogen, it is nitrogen limited. And then nitrogen productivity is capping out because then phosphorus becomes limiting. That's the next limiting nutrient. We add them both and we get lots and lots of growth. Now we look at the wet meadow, and we just have the forbs and the graminoids. We have a control, we add phosphorus, we get an increase in growth. We add nitrogen and we get an increase in growth. And with nitrogen, you're probably favoring the graminoids here with phosphorus. This is kind of you can call that not quite 50-50, 45-65. Kind of the same here. Here we're more like 70-30 in our species distribution. So each of these nutrients are limiting. We add phosphorus, we get more growth. We add nitrogen, we get more growth. And then that's kind of stopping because now we're, if you're adding nitrogen, then we grow, we get increased growth. And that stops because then it becomes phosphorus limited and vice versa. So you add both nitrogen and phosphorus and you get more growth. But what also happens here is a massive change in species distributions. And so the forbs become much less abundant, or they're contributing much less to the above ground biomass. And what's likely happening here is the graminoids, the sedges and grasses, are probably out competing and shading the forbs now that they're no longer nutrient limited, now that they have as much nitrogen and phosphorus as they want. So the addition of nutrients can both change overall primary productivity, but it can change the competition, right? So if an individual species that under normal conditions is nutrient limited and thereby not as strong of a competitor is now no longer nutrient limited, it can become a stronger competitor and change the dynamics of that patch. And so we have to think about that as we're adding all of this anthropogenic nutrients and how it's really changing the landscape by altering the normal fights for space and nutrients that would exist in a natural ecosystem. Now that we're adding anthropogenic nutrients, what would happen? And so in this example, you could say if through an anthropogenic source, nitrogen and phosphorus was added to a wet alpine meadow, what would you expect? And graminoids would be more dominant, and forbs would decline in population. So lakes were often limited by phosphorus and nitrogen availability. Many experiments, these are called limno corrals, are used to do these experiments. And sorry, here 1 second. And in freshwater marine systems, we can often estimate net primary productivity as a change in chlorophyll concentration or the number of phytoplankton cells. Where are we? So here's one of these limno corrals. So you have a lake, and then you basically have these like floating swimming pools within that lake. Then you can do manipulations. You can add different species or predators or different amounts of nutrients and see what happens as a way of having a number of experimental units, right? A number of replicates. And then you're also maintaining that natural conditions of light, temperature, etc., that you would have in a natural system. So often look at these questions like beaker scale experiments don't work, and so the next phase up from a beaker is usually things like cattle tanks, and so you have experimental stations that have these big either galvanized steel or black water tanks you can do experiments in, but that's also an artificial environment. So the next step is using these limno corrals, where we're placing these effectively floating swimming pools in lakes and then doing experiments. There are some examples where we can do whole lake fertilization experiments, which is really impressive. And this is really seminal work done by David Schindler. His son Daniel Schindler is a faculty at School of Aquatic and Fisheries Sciences at University of Washington. But here they were able to add nitrogen, phosphorus, and carbon to experimental lakes to see what would happen. And the phosphorus additions resulted in massive increases in cyanobacteria, photosynthetic cyanobacteria, showing that phosphorus was the limiting nutrient. Here's one image of that. And so here, this experimental lake region, they're able to do these large-scale experiments that you normally couldn't do. Like, I can't go down to Lake Padden and put a big effective drape halfway across the lake and then add a bunch of phosphorus to one half and not the other. But here you're allowed to do that sort of thing. And these large-scale experiments are really what's needed to understand some of these dynamics. In this case, the big question was around what is phosphorus doing in our systems. Actually, maybe I'll jump over and grab a couple of those slides because it's not really that relevant. In water quality, we talk about phosphorus at this time. This is what Schindler did his work. Experiment was in 79. He published it in 74. I mean, 69. The experiment was around 69. And phosphorus is a major component of soap, both like dishwashing detergent, laundry soap in particular, and then like hand soaps as well. And like anything, when there's some measurement of impact by an industry, there was a lot of slow movement to make changes around the phosphorus addition to these products, to these cleaning products. And so the issue was a question of scale, where scientists were able to measure adding phosphorus is increasing phytoplankton, but always doing it on small scales, water tank scales, beaker scales, etc. And so here's an example where, this next slide shows it better, where they're able to add carbon and nitrogen to one side of the lake, have this impermeable barrier here, add carbon, nitrogen, and phosphorus to the other side, and get a massive algal bloom. And here you can look at mean total phosphorus with mean annual chlorophyll, and you see a pretty strong relationship between the more phosphorus you add to the more chlorophyll you get. And so this finding was they were able to use this to change the way that phosphorus is regulated nationally, and now we can no longer have phosphorus in our dishwashing and laundry detergent, and there's some exemptions for that. But in general, this resulted in a understanding of phosphorus in the system, and leading to phosphorus being removed from primarily dishwashing detergent and laundry detergent, because it has a disproportionate impact on phytoplankton productivity, as many lakes are phosphorus limited. Now in streams, it's kind of interesting. You could think about the net primary productivity is often low in rivers and streams, and most energy is derived from terrestrial organic matter. Water flow limits phytoplankton growth. Most net primary productivity is from macrophytes and attached algae. And so the river continuum concept, which goes back to a couple of chapters ago, describes the increasing importance of in-stream net primary productivity as water flows downstream. And so one way of thinking of that is, if we had first, second, third, fourth order streams, and I were to add phosphorus to a first order stream, what would we expect the change in net primary productivity to be relative to adding phosphorus to a fourth order stream? Which one would I expect to have a greater magnitude of change? So which one would I expect a relative abundance of phytoplankton to increase, or net primary productivity in general? And one way to think of that is, well, how much of the energy in that stream, how much of the energy in that stream is coming from terrestrial sources versus in-stream primary productivity? And the order streams with more net primary productivity from the in-stream are going to have the biggest impact. And so think about the river continuum concept and what the addition of phosphorus would do along those different stages, and be able to articulate which ones would have the biggest overall impact. Now, stream sediment, what this is talking about is turbidity. There is no word turbidity. But turbidity in rivers can limit light penetration. Thus, clarity often controls net primary productivity. So as turbidity or water clarity, as turbidity increases or water clarity decreases, that's limiting light penetration. So that's limiting light penetration to both that benthic algae that could be growing and phytoplankton within the water. And so that can limit primary productivity in streams, as well as they can be nutrient limited as well. In marine ecosystems, the limiting nutrients can vary. Estuaries are frequently nutrient rich. That's where freshwater and marine water come together. And variation in net primary productivity is often correlated with nitrogen input from the rivers. Here locally, the Nooksack River is putting out a ton of nitrogen, primarily from from dairies and berries in the watershed. And this can result in algal blooms in dead zones. And we do have locally here not really a dead zone, but we do have very low dissolved oxygen concentrations in Bellingham Bay. associated with the mouth or the delta from the Nooksack River with dissolved oxygen values down to around 2 milligrams per liter, which is quite low. That's enough to support some clams and like infaunal worms, but not crabs or fish, things like that. So oxygen is limiting in Bellingham Bay, and it's associated with a seasonal pattern of phytoplankton blooms and die-offs, and so we tend to see these lows in oxygen sort of mid to late summer. Generally, the amount of nitrogen coming out of the Nooksack River is likely a massive contributor to that. In the open ocean, net primary productivity is mainly from phytoplankton, which is mostly nitrogen limited, which is why we see such massive growth around upwelling areas. So upwelling is the bringing of cold and nutrient-rich water to the surface, which is then scavenged by phytoplankton and used for growth, as it's frequently limiting. And it's really interesting that these picoplankton, smaller than a micron, so really quite small, contribute about half of our total marine net primary productivity. So extremely small phytoplankton play a really important role. And part of that's due to the surface area to volume ratio. And so small phytoplankton can survive better with a lower overall amount of nutrients, because the surface volume to area ratio, where if you have a much larger, you can think of like a basketball versus, I think I talked about this earlier, but a basketball versus like a golf ball. The basketball's got more volume given a certain surface area, so you need a higher concentration of nutrients to drive those nutrients across a concentration gradient, where in a smaller cell you don't need that. And floating seaweeds such as sargassum are important. Kelp beds and eelgrass are some of the most productive ecosystems on the Earth, and they contribute a lot to net primary productivity. But think about the ocean. How much of the ocean is kelp and how much is seagrass? Not very much. So the ocean does a lot of productivity, and it's really high in certain areas, but it's not the most of the ocean. Most of the ocean is a vast desert with crystal clear water and very little productivity. Of that productivity, it's primarily these little picoplankton. And it's kind of a famous experiment, but in the Southern Ocean, or the Equatorial Pacific, it's also referred to, it's been found that iron is limiting, and so in some of these systems, researchers will go out and we kept measuring a lot of nitrogen, but not a lot of primary productivity, and so it kind of gave this paradox of, well, normally nitrogen is limiting, but there's tons of it here. The water's warm, it's stable enough. We should have a lot of phytoplankton growth, but it turned out that there's another limiting nutrient, a micronutrient, and so over a series of experiments, of Iron X experiments, they added a ton of iron to the ocean, and it increased phytoplankton productivity up to fourfold of net primary productivity. These are in micrograms carbon per liter per day. And so this was kind of lauded at the time. I was like, oh, maybe we could fertilize the Southern Ocean and not have to alter any of our other behaviors, and use this as a way to draw down carbon dioxide. The issue is that if we did do this, if we did dump a ton of bioavailable iron in the Southern Ocean, yes, some of this carbon dioxide would be taken up by phytoplankton, but then it's returned to the atmosphere via respiration by zooplankton and bacteria. So meaning you get a phytoplankton bloom, and then the things that eat it also grow, and then they respire, and a good part of that carbon dioxide that was fixed returns back to the atmosphere. So it's not like every gram of phytoplankton equals a gram of storage. Iron quickly falls out of the photic zone, so it's tough to keep the phytoplankton going, to keep the growth going. So it's like interesting if like a quick like little shot and like a growth of phytoplankton, but to be able to sustain that is difficult, as well as what's going to happen to the benthos if we did do this and you've got increased flux of organic matter to the deep ocean? Could that be causing dead zones or hypoxic areas as well? So there could be additional ecosystem impacts. Okay, so yeah, we can look at remote sensing to get an idea of net primary productivity globally. And globally, the net primary productivity is about 105 pentagrams of carbon per year. It's not quite 50-50, but pretty close. It's taken up by terrestrial ecosystems versus the oceans. And this is kind of an interesting thing to think about, is what is the distribution of net primary productivity globally in both terrestrial and oceanic systems. And so just looking at oceanic systems, we tend to see increases at higher latitudes, and that's associated with upwelling. So upwelling is the big factor there, where we need to bring nutrients up to the surface to mid latitudes, bring nutrients up to the surface to support that primary productivity. And then on the terrestrial environment, it tends to be more around the equator, so more light, longer growing seasons, things like that. We also have a bump in the north, and so there's more land mass in the northern hemisphere compared to the southern hemisphere. So we tend to have more primary productivity in the northern hemisphere compared to the southern hemisphere. And you can actually see that annual cycle in the Keeling curve as well, that because of the differential of just the way that land mass is distributed around the globe, we have more land mass in the northern hemisphere compared to the southern. Okay, so that's mostly that for that one. Variation in net primary productivity in terrestrial biomes is associated mostly with leaf area index and length of growing season. Variation in net primary productivity in aquatic ecosystems is primarily related to variation in nutrient inputs. Look, I have a different shirt on. You could call that the second shirt I've worn during my lectures. Secondary production. So this is energy derived from consuming organic compounds produced by other organisms. Heterotrophs include archaea, bacteria, fungi, animals, and a few plants that we talked about in that section. We can define heterotrophs as herbivores, carnivores, detritivores, or omnivores. This should all be review. Herbivores eat plants, carnivores eat animals, detritivores eat detritus, dead things. And this is a cool one. So stable isotope ratios allow you to determine the source and trophic level of study organisms. And so you can compare the isotopic concentrations of a heterotroph compared to the food sources and figure out where it is, both on, if you want to think about it like the food chain, or what trophic level it's at, and also what the source of that food is. And I want to walk through this here in a second. And so, sorry, I had to go back to my first shirt just to be consistent, but I did wear two shirts. So in this study, this is pretty interesting. So they took Pleistocene herbivores and a species of cave bears that's been debated about what they're eating. The reason they had to measure Pleistocene herbivores is that where you are in the carbon space has to do with the ultimate source of that energy. And so different plants fix carbon differently, and depending on those type of plants, you'll have a different carbon signature. And so we don't need to get into that the mechanisms of that, but that goes back to like C3 versus C4 pathways. They fix carbon differently, so there's a different isotopic fractionation. So if we want to figure out what an organism was eating a long time ago, we also need to know what's the carbon signature of food from that time. And obviously we don't have the plant, well, we might, but generally we don't have the plant matter, a broad enough suite of the plant matter available to survey that. So what they did is they used an herbivore. And so they said, okay, we know this thing is eating plants, and so the carbon signature within it would be the same carbon signature of the plants it's consuming. In a modern study, we would sample the plants, get their carbon signature, and then get the herbivores to figure out, what portion of different plants they were eating. But nonetheless, in this example, they had a carbon signature and a nitrogen signature. The general rule in isotope ecology in nitrogen space is you are what you eat plus a mil. What a mil is, so this is delta, you'd say this is delta N15. Per mil is this measurement here. And so you would say that like this point right here has a value of roughly negative 21 per mil delta 13C. And then in nitrogen space, it's roughly 6 per mil delta nitrogen 15. And so here we've got a suite of herbivores and then these cave bears, which are unknown. Now they have the same carbon signature, which means the source of the carbon is the same. And you have to think back to the original source of the carbon. So if I eat a carrot and then somebody eats me, the source of that carbon is carrots. Or that's all I ate was carrots. So the source of my carbon would be from carrots. Whoever ate me, the source of their carbon would also be carrots, even though they ate me. But they would have about at least a 1 per mil higher amount of nitrogen. And so that's what we see here is we see the herbivores with the carbon signature of between negative 22 and negative 21 C. Unknown Pleistocene bears, we don't know what they eat. Same carbon signature, so their source of carbon is the same as these. And they're higher. They're, in this case, over a mil higher in nitrogen space, which means that they're a trophic level above the herbivores, which would imply that they're eating the herbivores. Now, they must have been using a different fractionation factor because they had... They assumed it would be eating about 58% meat, and then the rest vegetarian, which is true for like a lot of our bears, grizzlies even. They'll eat roots, berries, black bears more so with berries, but grizzlies will eat roots and things like that as well. So they're not purely carnivores; they're more of an omnivore, and they change their diet seasonally. So based on this, that's how they were able to figure out these cave bears. Had a large portion of their diet came from eating herbivores, and so it's pretty interesting stuff. I'm still thinking about how to put this on, but this idea of going up 1 per mil in nitrogen space is pretty interesting, and so being able to look at something like this, I won't necessarily query on them overall mechanisms, but knowing that trophic levels are differentiated in nitrogen space, I think is interesting. And so similar to how we look at net primary productivity, we can look at net secondary productivity, which is just what you eat minus what you respire versus things you pass that you don't eat. And so if you eat 100 calories, you respire 20 calories, and you excrete 20 calories. What did I say? 100, 20, 20. So there'll be a 60 calorie net secondary production. So it's basically like your mass. That's how we measure productivity is mass. And so net secondary production depends on the quality of food, digestibility, nutrient content, and physiology. Animals with higher respiration rates, endotherms, have less energy left over to allocate to growth, which is why things like ectotherms can survive on a lower diet. Snakes, things like that, they can eat one meal and be okay for a number of weeks, where that's generally not true for endotherms. And net secondary production in most ecosystems is a small fraction of net primary productivity. This is interesting. The fraction is greater in aquatic systems than terrestrial. So in aquatic systems, more of the carbon fixed by primary producers is consumed by secondary by consumers compared to terrestrial ecosystems. And that's a function of really, if you look at the standing stock of biomass in like marine versus terrestrial systems, you've got phytoplankton that only live for days to weeks. They're turning over more rapidly, where in terrestrial systems you can have trees that are 2,000 years old, right? They're not being consumed every year, where for phytoplankton, they divide quickly, grow quickly, and they generally don't live that long, so most of them are being eaten at a high rate. So it's really about carbon turnover is what we're looking at there. And most net secondary production is associated with detritivores, primarily bacteria and fungi. So everything dies eventually. A lot of it dies and rots, which, again, like think about, a tree. Like there's not that many things going out physically, like eating a giant tree. They'll die and rot away and then be consumed by detritivores. Same thing is true like in our lawns, other areas like that. You don't see that many herbivores out munching on things, but things are growing and dying and decaying. And so that detritivore and bacterial pathways are very important and wind up being the largest source of secondary production is associated with that of breaking down of dead or decaying plant matter. Okay, that'll wrap it up for chapter 20. I'll get chapter 21 up, and we will be nearly done.